Well, hi everyone. Welcome to today's live broadcast on CytoViva Hyperspectral Microscopy for Nano Drug Delivery. I'm Carol Ray, the Program Manager at CytoViva and moderator for today's event. I'll go ahead and start the introduction while we wait for the rest of our participants to join. CytoViva is a scientific imaging and instrumentation company that develops and markets optical microscopy and hyperspectral imaging technology for nanomaterials, pathogens, and general biology applications. Today, you will hear from Byron Cheatham, Vice President of CytoViva, who will provide an overview of the principles of CytoViva's enhanced dark-field optics and optical spectral imaging. Byron is a longtime technology development entrepreneur who's led the market implementation of CytoViva's optical and hyperspectral imaging technologies since the company's inception in 2005. During this time, he's overseen the successful implementation of CytoViva systems into hundreds of researchers' labs worldwide. So at this time, I will now hand the webinar over to Byron, and then afterwards, we will have a Q&A, and we can take it from there. So thank you, Byron.
Thanks, Carol. Thank you very much, and thank you for everyone for joining us on a Friday for this webinar. The webinar is being recorded, and we will provide a link to the recording for everyone after the webinar today, and we appreciate your participation. As Carol mentioned, I'm Byron Cheatham, and I'm Vice President at CytoViva. And my responsibilities are primarily around product development and market implementation of CytoViva's hyperspectral microscopy technologies. Today, we're going to focus on nano drug delivery related applications and their use with the CytoViva system. And so before we begin here real quick, just a quick overview of what we're going to cover today. The first thing we're going to do is just provide you guys a brief overview of CytoViva, the company, and the types of research applications that our technology supports, and the types of research institutions that acquire this technology and what they do with it very quickly. Then we'll transition into the technology, and specifically we're going to talk about the enhanced dark-field microscope optics and how they allow for label-free nanoscale imaging. And then we'll transition and talk about hyperspectral imaging and how it allows for the collection of optical spectrum in every nanoscale pixel of the image that's created by the enhanced dark-field optics. We're going to then transition and go through some very specific applications that are very specific to some of our research customers around the world and that they utilize. And we're going to focus on two things: characterizing drug loads and other functional groups that are on nanoparticles, and then also demonstrate how we can track drug-loaded nanoparticles in vitro cells, ex-vivo tissue, and how that can be done in a completely label-free manner. Can be done with live cells, fixed cells, and ex-vivo tissue that can be H&E stained or unstained as well. Okay. Afterwards, we're going to have an opportunity for questions and answers. So if you have questions, if during the webinar, if you could type those questions out in the Q&A, then we will cover those questions in detail until we've answered them to your satisfaction this morning. So thank you again. And with that, I'm going to start going through the process here. And I want to start by just making sure that everyone has a little understanding of our company very quickly. Our company has its roots in academic research laboratories. We were founded in around 2004 at Auburn University. We are in the Auburn University Research Park as a private company with a close tie and affiliation with Auburn University. Our enhanced dark field optics were developed at Auburn, and we continue to collaborate with them from an optics and spectral imaging perspective on a number of things. Since the development of our technology, it's been utilized and placed in almost 400 research labs worldwide. This just sort of gives you a sense of that. CytoViva as a company is a smaller company. We're a technology development company, and we primarily work with third-party distributors around the world who are licensed to exclusively distribute this technology in markets in Asia, Europe, and in North America. The technology has produced research on a very prolific level. And among those researchers, there's over 1,500 publications that are where the technology is referenced currently today. And there's some groundbreaking research that has been done in nanomedicine and nanodrug delivery, where the CytoViva technology played an instrumental role in that research that was done. And this just gives you a sense at a very high level of some of the types of research institutes that have acquired the technology, but more importantly, the kind of work that they've done with it. And I would say that in the nanotechnology realm, there are three primary areas where CytoViva has supported research institutes around the world. The first is in more pure nanomaterials development, synthesis, and the integration of nanoparticle technology into material science world, whether that's in photonics or whether that's in semiconductors or a wide range of things. Another area where CytoViva has had a significant influence and level of support is in the whole realm of nanotoxicology. And whether that's environmental nanotoxicology or whether it's human exposure, it's the idea that you can utilize this technology to quickly observe on your benchtop the interaction of nanomaterials and those nanomaterials interacting with a wide range of biological specimens and species. Okay, and then finally, drug delivery, which will be our focus this morning. I want to just give a real high-level overview of what the technology does. And these four bullets that I have here on this slide demonstrate this. Now, one of the things that I want to mention as we get further into the applications of the technology, we're going to actually get out of this PowerPoint deck, which can get boring real quick, I know. And we're going to actually open some Darkfield hyperspectral data files and conduct some live data analysis. And we're actually going to use the sample that I show here in this image on the right and some of the data that's representative of that on the left. But let's talk about what this technology does at a high level first. First of all, researchers that acquire this technology have the ability to optically observe nanoscale samples without any special labels or any special sample prep. You know, fluorescence, whether it's wide field fluorescence, epifluorescence, or confocal fluorescence, is a go-to tool, especially in biology, because it has tremendous utility and application. However, with fluorescence imaging, you can only see fluorescent labels. It's the essence of how confocal and epifluorescence works, which can be very limiting in a world where you're looking at engineered nanomaterials, which can be very hard to conjugate a fluorescent signal onto. And there can be tremendous disassociation of the fluorescent signal from the nanoparticles, and the chemistry of the fluorescence can fundamentally change the nanoparticle. And so it can really negatively impact what you want to really understand about these engineered nanomaterials. Plus, you can only see the biological environment, if you're looking at them in biology, if it's fluorescently labeled. So we're going to provide you the ability to be able to see fluorescent or non-fluorescent materials, to be able to see them in real time, and to be able to see them in stained, unstained, fluorescent, non-fluorescent biological material, okay? we're going to allow you the ability to spectrally identify and spectrally characterize nanoscale samples based on their scatter properties, their optical spectrum. Okay, we'll talk about what optical spectrum is as it would be different from spectrum from Raman, FTIR, and some of those sorts of things. And then we're going to allow you, as a result of being able to capture optical spectrum in every single pixel of the image, to conduct spectral mapping and do all sorts of other nanoscale analysis. So in this image of the right, we're looking at macrophages, and these are fixed macrophages that had been exposed to low-density lipoprotein lipids that had very, very small 2-3 nanometer gold particles internalized into the LDL lipids. And so you can see the macrophage cell here. This is an enhanced Darkfield Hyperspectral image. And this, there's no fluorescent labeling or any special sample prep on the sample. We see the macrophage. We can see these dots in the macrophage. Those dots are the LDL gold nanoparticle construct that the macrophage had internalized in the cell. And if we look at just the mean optical spectrum of the macrophage, we see this green spectrum on the left that represents that. If we look at just the mean spectrum analysis of the optical spectrum of the LDL gold loaded, we see this broad sort of amorphous spectrum, but they're very different. So that would allow you to do things like spectrally map these LDL gold loaded nanoparticles in these cells very quickly, very easily, very accurately. And I'm actually going to show a live demo of that pretty soon here. So with that, I want to transition and sort of give you guys a sense of what this technology looks like as it sits on your benchtop. And the first thing that you notice is that it looks like a fairly standard research grade optical microscope configuration. And it looks like that because that's what it is. And so this system is designed to take a basic research grade optical microscope, and we provide this complete system as a complete turnkey standalone system. And so we are actually a value-added reseller of the Olympus microscopes. And so we provide Olympus microscopes along with our optics and our spectral imaging integrated onto the Olympus system. So there are two very unique things about this Olympus microscope. One is the integration of our enhanced darkfield optics, which I'm going to talk about in great detail here in just a little bit, that mount onto the condenser mount of the optical microscope. And then in the camera mount, there are actually two camera systems. The first camera system is the spectrophotometer, which is made up of a diffraction grating spectrograph and an integrated camera. And then on the back of this dual port is a standard color optical camera, okay? And so you can position back and forth by moving this very quickly from hyperspectral image capture to high resolution color optical capture of video of nanoparticles and cell and tissue interacting, as opposed to collecting optical hyperspectral images of those same samples. Because sometimes there's a need just to capture optical images, and sometimes there's a need to capture the hyperspectral images to conduct the spectral analysis that's necessary for this. But this just gives you a sense that we're looking at a system that would look familiar to you, and it's the kind of thing that you would expect to have on your benchtop in your lab, as opposed to some system that would only reside in a core lab that's operated by specialists that you have to go down and utilize. So with that, I want to transition now and talk about some of the unique aspects of this system that allow us to optically observe and spectrally characterize at the nanoscale. And the first thing we're going to talk about are the patented enhanced darkfield microscope optics that were developed at Auburn University. But before I talk about these patented enhanced darkfield, everyone that's in Nano has heard of Darkfield, right? And Darkfield has some advantages in Nano. And so I want to explain how standard Darkfield works to give you some context as to how the CytoViva Darkfield produces a significant advantage. It's about a 10x advantage over standard Darkfield. With standard Darkfield on a standard microscope, light comes in from the back of the microscope for transmitted light Darkfield. It typically travels through the base of the microscope, and the base of the microscope has some lenses, collection lenses, field lenses. These are plastic lenses, typically in most all microscopes. And they're serving to try to organize the light before it comes to the base of the microscope to the field diaphragm. However, this can serve to create quite a bit of light loss just by the very nature of how it's organized. Then once the light comes to the field diaphragm on the microscope, it then has to travel through about 9 to 10 centimeters of air to reach the dark field condenser. And then to actually focus the light on the dark field condenser has to be moved up and down in the Z to focus it on the condenser. But then to focus the light of the condenser onto the sample, you have to once again move it into the Z. So you're always sacrificing focusing the light on the condenser, which is color illumination, to manually focus the light on the sample, which is known as critical illumination. That's a terminology that's typically used for that. So, and this is the way all major microscope companies have configured dark field optics, and it has some inherent disadvantages and relative to the power of dark field that's potentially available. And so the research group at Auburn University in the biophysics and vet med were using dark field a couple decades ago, and we're dealing with those inefficiencies and wanted to create an improvement in dark field, because inherently dark field has some advantages over traditional bright field microscopy and other methods, especially at the nanoscale. And so what they came to understand is that they could manage the light from the source illumination in a super precise manner, that they could improve the management of the light and bring more photons in a more focused manner onto the sample and produce a tremendous improvement in dark field optics. CytoViva developed the company around these commercialized optics, these patented commercialized optics. And in effect, what they do is they use a liquid light guide to connect the light via the light source directly. And we have a dark field illumination system that utilizes some collimating lenses and mirrors that are designed to change the geometry of the source light to more precisely match the geometry of the dark field condenser optics. And these are cardioid oil condenser optics, such that we can, without minimal photon loss, change the geometry of the light to match the geometry of the condenser. And that will allow us to put more photons onto the sample at a highly oblique, shallow angle and create more scatter of the nano sample into the objective without any additional stray noise from the light source being created. In other words, increase a significant signal to noise ratio. And to demonstrate that performance and how it works, this is an example of a paper produced a number of years ago by a research group that utilizes the CytoViva technology for microchip electrophoresis related work, where they're in effect chasing these 160 nanometer iron oxide nanoparticles in this microchip electrophoresis environment. And they have been utilizing the CytoViva optics for a number of years and getting really good results in terms of their ability to observe in the microchip electrophoresis environment these nanoparticles and how they're moving in that environment. But they happen to have dark field optics of whoever they had bought their microscope from some years ago. They had just acquired the CytoViva dark field optics for an existing microscope that they had. And they wanted to do an exercise, an experiment to see what the difference would be between those two. And you can see here in this experiment where they measured about a 10x increase in the signal to noise ratio, okay? And both signal and noise are important. With the CytoViva optics, we've created an ability to create improved signal of the scatter, whether that's Rayleigh scatter from a nanoparticle or me scatter from something bigger, an improved scatter signal, but also a decrease in noise And the noise is always being generated from the source illumination. So if we can increase that signal to noise, we can allow ourselves to observe the scatter from these nanoparticles interacting in other environments much more easily, much more quickly, and as you can see here, with a much higher degree of specificity. And so once that had happened, we knew that we needed the ability now to be able to conduct some characterization of these images, these very high signal to noise images of nanoparticles in a wide range of environments. And so we worked, well, we actually had, there was a research customer at Purdue University that had been dabbling with optical hyperspectral imaging for number of years. And they were looking at gold nanorods and cells, and they had a home-built version of that. And we collaborated with them to understand what they were doing. And that led us to the development of some highly effective and proprietary commercial hyperspectral imaging technology that we have that was developed specifically for nanoscale related work. And I'll explain really quickly how it works. It is a scanning, diffraction grading hyperspectral imaging system. And it allows us to capture the visible near infrared, or we can also capture shortwave infrared optical spectrum in every pixel of the image. And so this is a scanning system. And so we can scan a sample. The image that I showed you of the macrophages and LDLs took just seconds to capture that little small area that we were looking at. In the visible near infrared, which is where about 90% of engineered nanomaterial researchers are working, we're capturing all of the spectrum in every pixel from 400 nanometers to 1,000 nanometers. If you're working with materials that produce an optical spectrum and more completely in the near infrared range, and for example, there are some nanorods that produce optical spectrum in that range, we can capture with a shortwave infrared spectrograph and an InGas camera, we can capture hyperspectral images from 900 nanometers to 1,700 nanometers. These are nanoscale pixels. Pixel sizes can be as small as 100, 128 nanometers at the highest magnification. Now, that's not the limit, though, on the scatter detection that you can see. Because, for example, a 50 nanometer gold nanoparticle is going to have scatter properties and scatter size that well exceed even this 128 nanometer pixel. It will probably have scatter properties that will equal 200 or nanometers or more, especially with these enhanced dark field optics, which produce very high scatter efficiencies. So that gives us the ability to detect metal nanoparticles, and numerous publications have demonstrated this, down to the 10, 15, 20 nanometer range, and measure their optical spectrum. Things like metal oxide nanoparticles in the 20, 30 nanometer range quite easily, polymeric nanomaterials in the 30, 40 nanometer range. Lipid-based nanomaterials has the detection capability in and around the 50, 60 nanometer range to give you a sense. Now, all of these nanoparticles scatter light with different efficiency based on their chemistry and other variables. So not all nanoparticles are the same. And of course, you understand that as it relates to how they interact and scatter light. But that gives you a sense of the type of detection that we can do. Now, the spectral data from 400 to 1,000 nanometers in the Wiener is very high. Spectral resolution, not spatial resolution, but spectral resolution is really less than about 2 nanometers. And the most interesting thing about this data is that it's all presented in spatial context of an image, of a very high resolution image. The data is presented as a red, green, blue image for your eyes, but you have all of the spectral data from 400 to 1,000 nanometers with 2 nanometers of spectral resolution in every pixel. And that gives us the opportunity to conduct all sorts of detailed quantitative analysis. I'm going to show a simple example or two of that as we move on here. This, though, demonstrates the power of proper optimized dark field optics for hyperspectral imaging at the nanoscale. This is the identical 100 nanometer particle, 2 hyperspectral images, one captured utilizing standard off-the-shelf dark-field optics that you would get from a microscope company on the left, and then the exact same nanoparticle hyperspectral image captured using CytoViva's enhanced dark-field optics. The signal-to-noise ratio is intuitively obvious here, and that produces a dramatic difference in the optical spectral signal that we're able to capture in terms of its effectiveness. And it also can increase the clarity of the preciseness of this optical spectrum. So you see this optical spectrum that's really clean has a peak at about 650 of this same pixel of the gold nanoparticle. And with the stage and with registration, we can always go back to an identical pixel area with very high effectiveness on this sample where it wasn't moved off the stage. The only thing that was changed was the dark field optics. Here we see a noisy signal. It has a peak at about 600 nanometers. And the reason why it has a shift is because of the noise. The noise is a function of the illumination optics, which influence the signal a lot more in this image, and you can see it. Now, there's a way to normalize the data out to do what's called a lamp normalization, and you might want to do that with either of these images that you capture, but that just gives you a sense of the improvement in the signal to noise. So with that, I'm going to transition away from talking about the technology and the optics, and I'm going to transition to how it's used in the research world. Among these hundreds of researchers, many of whom are involved in nano medicine and nano drug delivery related applications. And I would encourage you, if you have any questions about the technology as I've laid it out, please type a question and we will be more than glad to answer that. Also, if you have more questions, you're welcome to come back to us later at CytoViva, info@cytoviva.com, if you want to have a more detailed discussion about some of the questions that you have. So the first thing I'm going to do is talk about identifying functional groups on nanoparticles. So, you know, one of the most difficult things to try to do as you have engineered nanoparticles, whether it's polymer nanoparticles or lipids or liposomes, is to try to load functional groups, specifically drugs, on or in, and to understand across a sample what's my efficacy of the loading. Do all of my nanoparticles have the functional group or drug load on them, or just do a few? And how does that look in the context of where those drug loads or functional groups are on the nanoparticles? This technology can help you do that. And I want to show an example here in the PowerPoint, but we're actually going to go to a live image of this, and I'm going to demonstrate this to you a little later. So these are polymer nanoparticles captured with Darkfield Hyperspectral Imaging in solution. Nothing changed from them from their native endogenous state. They were put on a slide, they attached to the slide, and there's two differences between these two guys. This is a negative control sample. This is a vehicle of the polymer nanoparticles. This is the identical polymer nanoparticles, but cisplatin drug has been added to these polymer nanoparticles and to their surface. Okay, and so this is some representative spectrum of the nanoparticles with the API, with the cisplatin small molecule drug. This is an example spectrum of the nanoparticle without the API, the vehicle, the negative control. And so based on this information, we're able to collect representative spectrum of this image. We could collect all the spectrum of this image as a spectral library. And then we could filter that spectral library against every pixel of the negative control to create a spectral library that's unique to the elements of the API loading on the polymer nanoparticle. And so once I've got that spectral library, I filtered it effectively against the control. I now can utilize other algorithms. There's a supervised classification algorithm known as spectral angle mapper that we use a lot in hyperspectral imaging. And utilizing the spectral angle mapper algorithm, we're able to map every pixel in this image that has unique spectrum relative to the negative control. And the only difference between these two samples is the loading of the API, the cisplatin small molecule drug. And so you see that we're mapping in red on the surface of these nanoparticles because this cisplatin is not in the nanoparticles, it's on the nanoparticles. And we must remember these nanoparticles are spheres, three-dimensional objects, but we're looking at a two-dimensional spatial image of these, okay? So we're only going to map these nanoparticles, this API on the outside of these guys, and you can see that here. And so what I want to do now is I want to transition and actually show you how this looks with a live hyperspectral image, okay? And so you can now see on my desktop, I have a hyperspectral image file of the drug-loaded and a hyperspectral image file of the vehicle negative control. And we're looking at representative spectrum of these guys here, right? As I move around, it changes, and it changes because, you know, the API is not consistent with it. It's different, and it changes, okay? So representative spectrum here, you're going to see, you know, less change with this guy because it's just a polymer nanoparticle, okay? However, when we went in and captured large areas of spectrum from this sample, okay, and then we filtered that spectral library against every single pixel of multiple negative controls, not just one, we were able to create a spectral library of all of the unique spectral elements that are due to the loading of the API onto the nanoparticles. And then based on that, I'm able to spectrally map every pixel in the image that's unique to the spectral loading. And you can see that the spectral loading shows that the API is around these guys, okay? You can see it's around them in various places. And then as I turn that off, you can see as you go to different areas here where that loading is, where it's really different, right, in the spectrum, okay? And if I turn it off again, that gives you a sense of how it's loaded around. And we can even get a class distribution that will give us a sense of the relative loading that we have in this image relative to the total number of pixels that are available, some quantitative data that we can get on that. And so that is one example looking at loading of functional groups on nanoparticles and how that works. Just to demonstrate a couple of more examples, here we're looking at liposomes, and these are liposomes that are being utilized for gadolinium delivery. Gadolinium is a drug approved for MRI-related contrast studies. The liposomes can deliver that much better. If you look, the liposomes have very identical spectrum, except when they're gadolinium loaded, it has a peak at about 750 nanometers, 770 nanometers. That's quite unique for the gadolinium loading, which allows the researcher to understand the efficacy of the gadolinium loading in the liposome in that example, to show an additional example of drug load effectiveness. With that, I'm going to move on and show just a few examples of drug-loaded nanoparticles or functionalized nanoparticles in vitro cells and ex vivo tissue. And we're going to start off with in vivo cells. The first I'm going to show is we're going to look at liposomes. And a lot of emphasis on liposomes and polymer nanomaterials because they're probably, in the world of nanodrug delivery, they're probably the engineered nanomaterials that have received you know, the highest adoption for potential clinical application. That's not to say that, plasmonic nanoparticles, and we're going to, I think, show an example there of plasmonic as well, have great efficacy too, and you know, work quite well. But if we can work with lipids, we can certainly work with plasmonics very well. So here we're looking at DOX-loaded liposomes, and we're looking at a representative optical spectrum of the doxorubicin-loaded liposomes. And you see this peak, this really sharp peak in the spectrum. DOX is known to have a natural fluorescence, and it's known to have a natural fluorescence in the almost in the late green, yellow, early red area. And so we see this sharp peak that appears consistently in these doxorubicin-loaded liposomes that show a peak optical spectrum here at about 585, 590 nanometers. That's representative of the DOX load that's in the nanoparticles, We see other spectrum here in this spectral curve, optical spectral curve that's related to the lipid characteristics as well. Here we're looking at cells, and these are cells that have been exposed to the DOX-loaded lipids. Now, liposomes are not going to appear to the naked eye to have any real optical difference versus cell vacuoles, endosomes, organelles in the cell. So you're not really going to be able to see them just optically. But based on the optical spectrum, especially the optical spectrum of something as unique as the DOX is loaded there, that's unique to the cell structure. You're not going to find that in the cell structure. So you can build a spectral library of DOX-loaded liposomes in a cell. You can filter that against the negative control or vehicle cells and create a unique spectral library, again, for unique spectral mapping. And what we see here, there's a ton of DOX-loaded liposomes on the cellular membrane and also around the nuclear membrane in this case, okay? And I would suspect that this is probably an application where the DOX is released and probably will be preferentially crossing the nuclear membrane. I don't know specifically, but this gives you a sense of where those DOX-loaded liposomes were in the cell vis-a-vis the spectral mapping that we're looking at here. I want to go back now, and this is the exact same sample that we were looking at a little bit earlier. The low-density lipoprotein gold nanoparticles in the macrophage groups. And so here we're looking at the full image of this, and we can see LDL that's loaded in the or taken up by various macrophages that we see here. In this image, we're looking at spectral mapping of those guys, and we see really nice spectral mapping. Anywhere we see the LDLs present, we get a class distribution that gives us quantitative information relative to the mapping in the image that we're looking here, okay? And that gives you a sense of where those guys are. So one other thing that we wanted to look at real quick is looking at tracking of drug-loaded medical grade CNTs, and we're going to transition and look at these in ex vivo tissue. So there is the opportunity to look at ex vivo tissue and conduct the same sort of analysis that we're doing, okay? So here we're looking at tissue that have medical grade, really small nanotubes that are again loaded with a cisplatin chemotherapy, okay? And so this is the spectral library that's representative of the unique aspects of the nanotubes with cisplatin chemotherapy loaded. You can see some white punctate areas here. That's going to line up with a lot of the spectral mapping that we're going to do with this in just a little bit. And here we're doing mapping of two things. We're doing mapping in the red of the nanotube chemotherapy, and then only, okay? And then nanotubes alone are in green. So what we see is that in the red, that we see lots of release of the active ingredient, the pharmaceutical active ingredient, and in green, we're looking at the nanotubes alone to demonstrate that. And here is the spectrum of the nanotubes alone. Here's the spectrum of the nanotubes with the cisplatin chemotherapy to demonstrate that the functional groups added to these guys really change their optical spectrum. And so with that, I'm going to get rid of some of these files that I have open here, and we're going to actually bring in another sample into this image to show you how this looks. Pardon me while I bring this in very quickly. This is the macrophages with the low density lipoproteins that have the gold nanoparticles. I'm changing the enhancement filter, the way these are presented, so that, again, this is all spectral data in every pixel of these of these images, right? If I click on a z-axis profile here, right, and I click on an area, you can see how we get different spectrum. I'm actually going to bring this in so that we can see here in a zoom area what the spectrum is, and we see the spectral response of an LDL loaded gold nanoparticle and how different that is. And you see a couple of things. You see difference in the spectral response, intensity, because gold loaded low density lipoproteins are going to have a different intensity and different optical spectrum from the cell structure itself, and different peaks, okay? But what I want you to be able to see here in this image is, as we built the spectral library, filtered it against the negative control, and conduct a spectral mapping, that we're able to spectrally map those low density lipoproteins in this cell structure very effectively, and give you a sense of how that works again. Okay, so with that, that's the end of the formal part of the presentation that I had today to demonstrate the effectiveness of this system to allow us to observe nanoparticles interacting with ex-vivo tissue, in vitro cells, and to conduct spectral analysis to demonstrate the presence of a drug load on nanoparticles and those nanoparticles in a drug load in those cells and tissue. And Carol, with that, I think it would probably be appropriate to open up the discussion. One of the things that I do, and it wasn't on here, but I'm going to add it. If you have a question that would be longer than what we can answer, I am going to also include our email address here that you can send an email to us on. But Carol, with that, I'll turn it back over to you, and we'll start on questions that we have, and maybe some questions in the chat as well.
All right. Thank you, Byron, for that very interesting presentation. I know everyone enjoyed it. So I will look through this chat. So the first question we have, what is the best fixative for cell and the best mounting to avoid noise? Right.
Well, I'm going to proclaim that I'm not a cell biologist, and I'm not an expert on fixation of cells. I would say two things: is that we've not seen that that fixatives really create a lot of noise that impacts the spectral analysis or the imaging. Okay, so a wide range of fixatives will work. There is the ability to look at live cells, and there's ability to do extended live cell study. So we work with an organization called CytoViva that prints a live cell chip that allows you to actually grow live cells on the chip, and to introduce nanoparticles with a drug load, and over time and in real time observe the uptake and impact of the cells on the impact of the nanoparticles on the cells. So you have the ability to do live cells and fixed cells. I don't know that I can say that one fixative is bad and one is good. Sometimes it actually comes down to proficiency of the grad student and their ability to do a good job in the fixative process. Sometimes that matters more than the type of fixative that you're actually using, I would say. And then also with regard to tissue, sometimes when you're working with pathologists, they really want to see an H&E stain on the tissue. Tissue can be looked at without stain or with stain, and nanoparticles can be identified in that environment. Okay. Now the stain is going to influence the optical spectrum, but a nanoparticle with H&E stain has a different optical spectral response than a tissue area that's H&E stained. Okay, even though the stain influences both of the spectrum. So you have the ability to look at ex-vivo tissue that's stained or unstained, and fixative is, differences in fixative is not a big issue.
Okay, thank you. Also, how did you take the HSI of only the API?
Well, we didn't. So we did not look at only the API. We look at, you can, okay, a high concentration of dots is going to create some scatter, okay? And we have imaged free dots in a high concentration, in a high creation, okay? Because it's not colorless or odorless. Now the cisplatin, I think what we're only looking at is the optical spectrum of the cisplatin interacting with, in this case, the polymer nanoparticle or the carbon. So I don't know that cisplatin was looked at on an isolated basis, and I don't know that that's possible. But what we're really looking at is the vehicle, the polymer nanoparticle, and the polymer nanoparticle functionalized. Because what's really happening is that the small molecule drug is changing the way that light interacts with the nanoparticle. Okay, it's changing the way that light is absorbed and reflected by the polymer nanoparticle as a result of its presence on it. The gold nanoparticles in the bilayer of a liposome change the way that the liposome is absorbing and reflecting light. The plasmonics interact with that. So these small molecule drugs do absorb and reflect light and do change dramatically the way that a nanoparticle will absorb and reflect light. And I don't profess to be an expert on the chemistry of cisplatin or doxorubicin and these kinds of things to understand how it changes the optical spectrum. I just know that it does on a fairly consistent basis. And I've worked with researchers that have explained it to me before. I won't try to go through the chemistry of that today, but you know, the folks that understand the chemistry of these small molecule drugs can explain that better than me.
Okay. To I guess add to that, Ara asked, then how do you know that that's actually the spectrum from the API and not from the polymer?
Well, so you have to look at just like any, first of all, it's a controlled experiment, okay? And so in the controlled, all of this is a comparative analysis technique, and it's a controlled experiment. But one of the things that you want to do is you want to look at not just one sample, but you want to look at 3 API-loaded nanoparticle constructs and capture hyperspectral images of it, and 3 vehicle controls. And you're looking for consistency across that. If there's only one variable change from the negative control to the API loaded, there's only one thing that can cause the change in the spectrum. And so what we can do is collect optical spectrum of all of the optical spectrum that's unique to the loaded. And then we can filter that spectrum against every pixel of all of the negative controls. And that comparative analysis technique is going to produce the optical spectrum that's different. And that's a classification, that's a supervised classification algorithm that you can set to operate with 99.5% confidence to know that everything that you're mapping is unique to the one variable you changed. And that one variable can only be the small molecule drug in that example. So that's how you do that. Now, also, no technique provides the complete answer to any scientist's question. And I would suggest that you would further clarify that. And there are a number of ways that you can do that. One that comes to mind, for example, is ICP mass spec. You can take that sample and make a confirmation with ICP mass spec as well. And it would even provide you more quantitative data with regard to that. The difference is ICP mass spec is a destructive sample technique. ICP mass spec doesn't allow you to understand how the API interacts with the nanoparticle. It'll just tell you if it's there or not. But that's a good correlative way to try to understand what you're seeing in this technique, for example, as a cross-check. We would always recommend a cross-check with additional instruments that provide complementary data in a different kind of way.
Okay, thank you, Byron. Next question. Can hyperspectral detection allow for a semi-quantitative assessment of a particle presence in a complex image, so containing other materials and particles?
It can, and there have been examples we do a lot of work in material science where there may be two different nanomaterials present that are two different that they're looking for, you know, gold and silver, for example. We've had some applications where there were gold and silver nanoparticles, and so they're going to produce different optical spectrum. The key is that you know, these are controlled experiment in environments every time, and they have to be. So if I'm looking, let's say I have a complex polymer substrate, and I'm for a transdermal drug delivery process, right? And I have nanoparticles in the polymer complex, and I want to see these nanoparticles, or I want to see these nanoparticles with a drug load, or maybe I have two different types of nanoparticles. As long as the experiment design is set up to allow us to identify each individual element that we want to, the potential exists to use it. Now, it doesn't answer every question for every type of sample that you might have. I don't want to represent that because that would certainly be inaccurate. But as long as the experiment design is set up properly, we can have a high degree of confidence. The other thing is that you can go very detailed on the spectral analysis, and sometimes you really have to. And on some very complex samples, it requires a deep understanding of all of the analysis capability that's present. One of the things that we've done, one of my colleagues, Jamie Ertz, Jamie is one of our chief developers and leads the software development process along with some others. And Jamie has developed some very extensive libraries of videos where he demonstrates how to use the technique with samples exactly like this to try to conduct detailed spectral analysis to answer specific problems. And we probably spend most of our time with our research clients around the world working with them on refining different analysis techniques. And we'll even go so far as to trying to work with a number of research customers that may be creating a new analysis technique or trying something new with some new samples to see if we get the same data result that they get, since we have such a deep knowledge, and we do that on a regular basis and enjoy that.
Wonderful, thank you. I have noticed there's a few questions about sample prep and if there's any information for, you know, sample prep online, and I am putting in the chat the link to our webpage that gives you some detailed information on optimizing your samples. And next question that is regarding sample prep. Do we need the nano to be in suspension, or can it be analyzed, dried after being brushed in a glass slide, for instance?
Absolutely. So the one thing about hyperspectral imaging, full hyperspectral images, is that your sample cannot be moving. So if I put 5 microliters of solution containing a concentration of nanoparticles on the slide, you know, you're going to see those nanoparticles moving in Brownian motion, and depending on their size, of course, they can move really rapidly if they're small. And so nanoparticles moving in Brownian motion don't make for good hyperspectral images because they're moving. You would see streaks because it's a line scan method. However, you can allow those guys to settle down on the slide. They're going to eventually attach to the glass. Some of them will. You can allow it to dry. You can do spray coating of the nanoparticles onto the slide. You know, even live cells, you know, even adherent cells on the glass, there's going to be a little bit of movement. That's okay. You just don't want a nanoparticle zipping across a 400 nanometer field of view while you're trying to capture a hyperspectral image randomly in Brownian motion. That doesn't work very well. But one of the things that we work with you on is if you had a system in your lab, is the different techniques for how you can put nanoparticles on the slide, how you can do your cell culture, conduct your cell culture. And it's an interesting thing. There's been somewhat of a fraternity of users in the nanospace that use this technology, and a number of them communicate with each other. We oftentimes will put a new user group in touch with a more experienced user group that we know has experience relative to sample prep on these techniques. We're much better at the imaging than we are sometimes the sample prep. most of what we've learned from sample prep, we've learned from our research clients around the world who have conducted a lot of trial and error over the last 15 years.
Okay. Another from Aura. How did you map 2 different spectrum materials and run it in an individual sample?
So the way you do that is you would create a spectral library, a filtered spectral library of both elements. So 2 different spectral libraries. And then what we typically do is we put both of those spectral libraries into a single spectral library. You know, spectrum 1 through 10 equals, spectral library A, spectrum 11 through 20 equals spectral library B. And then we color code A green and B red, and we map it in the same image in that format. So that's a deep dive technique that we could share with you, as part of the training that we would conduct if we were installing a system at your lab and showing you how to use it typically.
All right. How did you run a control spectrum and have it as unclassified?
So you create a hyperspectral image of your negative control, right? And then you conduct, there's a supervised algorithm. Again, it's part of the supervised algorithm. It's called filter spectral library. And so as I build a spectral library for my positive control, I then run filter spectral library algorithm that's in the software against the negative control. And so that's a really deep dive question, which would sound like maybe it's coming from an existing user with a question. To really go into the details of that, we'd probably need to do that more in a one-on-one basis. But there is a process for doing that. Yeah.
Okay. Can the sizes of nanoparticles characterized using this technology?
This technology is not a nanoparticle size analysis system. You can see nanoparticles, and you can see that if a nanoparticle, if you had a polymer nanoparticle that was 60 nanometers and you had a polymer nanoparticle that was 150 nanometers, you're going to see they're bigger, right? Just because of their scatter properties. But this is not a tool designed to give you accurate data on those size differences, okay? But it is something that's going to, nanoparticle tracking analysis, dynamic light scattering, those are all tools that are specifically designed to give you nanoparticle sizing and count related information. And I would suggest that that's your best way to get that information. However, those techniques sometimes can be difficult to calibrate and get accurate. We oftentimes find that when we qualitatively look at nanoparticles, that NTA, nanoparticle tracking analysis, or DSL, have some sort of bias in them, with bigger particles or whatever that can, when we see, wow, this doesn't look like what dynamic light scattering said, that will often lead you to the electron microscope, which is the ultimate method in my mind to try to understand nanoparticle size distribution in some cases. As we all know, that's a highly laborious method, but it is one that works. I will say on plasmonic nanoparticles, though, as plasmonic nanoparticles get bigger, their optical spectrum redshifts in a fairly quantitative manner. So, for example, if I have 50 nanometer gold particles and I want to know if I have one or if I want to have an aggregate, we know that a 50 nanometer gold particle in a refractive index media of water is going to have a reflectance optical spectrum of about 560 nanometers. And if there's an aggregate, that's going to redshift. So you can get some data, and if you understand your sample and understand its optical spectrum pretty well, especially with plasmonic nanoparticles, you can get some really insightful data relative to nanoparticle size, especially plasmonic nanoparticles, okay? And you can clearly see dispersed particles versus aggregates here. But any size information, for the most part, is qualitative. And it should correlate pretty well to what you see in your dynamic light scattering or NTA. If it doesn't, you might want to go to the ultimate nanoparticle sizing tool that's, of course, laborious to use, the electron microscopy.
Okay. Let's see. Can you look at and differentiate cisplatin or gadolinium on gold nanoparticles as opposed to liposomes?
You can, yes. And so we've looked at on gold nanoparticles, the surface plasmon resonance, of course, is so intense. But whether it's doxorubicin or we've seen differences in small molecule drug added to gold nanoparticles, just like we have with polymers, and because we're going to change how that surface plasmon resonance is measured in effect. So yes, and utilize, and usually it's a super obvious thing. I will say we've not looked at a lot of drug loads on plasmonic nanoparticles like we have with polymers and lipids, primarily because polymers and lipids are what we see a higher prevalence of in the research world, because that seems to be what the clinical world is more interested in. But we're seeing more and more plasmonic as well as time goes on.
Okay. Can you distinguish a protein incorporated in the nanocarrier or attached to the surface in a cellular environment?
Say it one more time, Carol, please.
Can you distinguish a protein incorporated in the nanocarrier or attached to the surface in a cellular environment?
So if you functionalize nanoparticles with certain proteins, there is the potential to do that. Now we're getting into some gray areas, I would say. For example, if you put, a traditional molar weight of PEG, polyethylene glycol, on a nanoparticle to make it more biocompatible, okay, we've been able to actually measure changes in optical spectrum based on different molar weights of polyethylene glycol added to nanoparticles. Plasmonic nanoparticles come to mind. So that potential exists. Whether or not you can see those in a cell structure, I don't know that I have any that come to mind. That's a lot of this stuff is very assay specific as to how well it works. I will say that. I mean, I don't want to represent that every nanoconstruct and every environment works, because it doesn't. That would not be appropriate to say. But you can go out into the literature and see if what you want to do exists. If you don't see that readily, you can contact us and get that information if we have it. If we don't, and that information would be relevant to you potentially thinking about adding this technology to your lab. Oftentimes we can do some proof of concept work for you out of our demo system lab to answer that question, and would be pleased to do so.
Thank you. There's just a few more.
Yes.
I was wondering if there is any protocol for running a control mapping versus the spectrum from the nanoparticles.
A control mapping, say that again, just to make sure I understand it.
Is there any protocol for running a control mapping versus the spectrum from the nanoparticles?
I don't know that I understand that question effectively enough to answer it. So maybe that might be a question better answered in a discussion offline. I'm not really sure that I completely understand it. My apologies.
Okay. Let's see. Would you say if gold nanoparticles of 100 nanometers are used, can we have a difference between aggregates or single particle spectrum?
Absolutely. To the point that I just made earlier, is that plasmonic nanoparticles, 100 nanometer gold particle is going to have plasmonic nanoparticles have very distinct optical spectrum due to the surface plasmon resonance. And if you put three together, it's going to change the spectrum. There's going to be a broadening of the spectrum, and there's going to be a shift in the peak. And that's pretty quantitative stuff that happens. That's really quantitative stuff that happens. And you'll see a difference. 300 nanometer gold particles aggregating are going to produce a different spectrum and are going to look different than a single. And we've also done a lot of work in the dimer world where, 40 nanometer or 60 nanometer gold nanoparticles are purposely placed, nanometers apart. And that creates an amplified effect. Dr. Naomi Hallis's lab out of Rice University has done a lot of work with this, where they look at specific dimers, and hers were silica gold shells, but it doesn't matter. And you're going to get really specific optical spectrum based on how close those 200 nanometer particles are apart from each other. There's an amplification effect that takes place there. And again, I'm not a physical chemist and don't want to try to represent what's happening there. But we do know that you can get differences in optical spectrum, plasmonic nanoparticles of a certain size and of a certain distance apart. So this technique can be very quantitative in those types of studies.
Thank you. Now, is there some kind of spectral library of nanos in various solvents that exists?
There's not, and we get asked this question all the time. The thing about nanoparticle synthesis at work and the use of nanoparticles in a wide range of environments, almost everybody in every basic research lab is doing something a little bit different, right? That's the whole nature of basic research. And so the idea that we can create, an optical spectral library of nanoparticles, and it's going to be something that can be ubiquitous is not possible, because there's a slight variation on the theme for almost everybody we work with. But it's super simple to create these spectral libraries for whatever experiment that you've got. That's something that we do during the training process. And one of the things that we often do during the training process is we recommend that you have an experiment, something that you want an answer to at the training. And we'll actually focus the training around that. So two things happen. You're really interested in the training because it's solving a problem right in front of you. And so it's really easy to pay attention. But at the end of the day, you've already got some data relative to whatever problem you're trying to solve right there then too. So that's part of what we do.
Mm-hmm. Last question. Can we change the scale bars from micrometer to nanometer with the right dimensions?
So the scale bars always we typically recommend you do them in ImageJ. They're not done in the hyperspectral image analysis software. And you can make scale bars whatever you want them to be in ImageJ, as far as I know and understand.
Okay, perfect. So thank you everyone for all the great questions. And thank you, Byron, for answering them so well.
Very good. Very good.
Yeah.
Thank you guys very much. We'll conclude the webinar here. This will be recorded. We'll send out a link to the recording to everyone, I guess, Carol, right?
Yes, everyone that's registered. So you'll get a link to this recording. So
awesome. I hope everyone has a nice weekend, and thank you for attending.
Yes, thank you.